My name's Dr. Ian Coombs. I am the admissions tutor for the pharmacology degree here at UCL. And what I'm going to go through today is basically give you an introduction to the pharmacology degrees that we offer here at UCL. So this is a BSc and an MSci. And also talk a little bit about some of the pharmacology research we do here at UCL. So UCL is a really, really strong research centre within the UK, and our department is part of that research effort. And some of the research that I'll specifically talk about done in my lab will be research that you can potentially get involved in as students on studying pharmacology at UCL. And joining us today, we have, I think, four students from the pharmacology degree who will also be around to answer any questions you have, either dropped in the chat as we go or saved up for the end. So hopefully we'll have about 20 minutes to answer questions once I finish. Okay, so specifically what we're talking about in terms of these degrees, in terms of UCAS, is course B210, which is the Pharmacology BSc, and B211, which is the Pharmacology MSci course. And I'll go through the differences between those two as we go. And I guess the most important question, because you may be looking at various other kind of types of degree, other subjects that are in this kind of area, what actually is pharmacology? So pharmacology is the study of the mechanisms of drug action. And these can be obviously both therapeutically useful, ones we use to treat illnesses, but also recreational drugs and drugs of abuse. But it's more than just the discovery of artificial molecules to stimulate the body. Trying to understand more about the way that these receptor molecules normally work will give us a much better idea about how the drugs that we try and design will affect the body. So it's more than just about drug discovery. It's about working out and discovering more things about the receptor proteins themselves. But a really important distinction to make is it's not the same as pharmacy. So pharmacy is a vocational degree. Pharmacy is what you study if you want to become a pharmacist. So if you go to the doctors and you get a prescription for a medicine, you will go to the pharmacy, and the pharmacist is the one who will make sure you get the right amount of your drug, make sure it's appropriate for the condition that you've been given it for, and also tell you useful bits of information like take this with food or take this at night, things like that. But the actual discovery of the drug that they are doling out, that was done by pharmacologists. So pharmacology is a science, and a science of discovery. And so a slightly more kind of complicated definition of pharmacology could be this. So if physiology is concerned with the function, anatomy with the structure, and biochemistry with the chemistry of the living body, and pharmacology is concerned with the changes in the function, the structure, and the chemical properties of the body brought about by chemical substances. So this really does give an indication of how pharmacology is at the intersection of many different divisions of biology and chemistry. It's a very, very interesting and fascinating subject. Okay, so if you decide to study pharmacology, and I hope you do, then why would you choose to study at UCL? Well, UCL is one of the very top universities in the world. So we've been ranked in the top 10 for a number of years in the QS World University Rankings, and we've been 9th the last couple of years. And just for comparison with other, some American universities you may well be familiar with, Harvard in that time was 4th, Yale was in 16th and now in 23rd, and Princeton was in 17th and now in 22nd. So we really are right up there with and better than some of the other leading universities in the world. In particular, why you might choose to study pharmacology at UCL? Well, UCL is ranked 3rd amongst world's top universities for pharmacy and pharmacology. So we were 4th last year, this year we're 3rd. And this compares with other places such as Cambridge in 6th, and Yale and Imperial were joint 17th. So we really are a really, really top place to study this science. Okay, and why is pharmacology at UCL so strong? Well, one particular reason that we're very proud of is that we're really strong on practical sessions. We believe that getting you into the lab And actually having your hands on experiments is the best way to learn how to become a pharmacologist. And we spend much more time in the laboratory than you're likely to find at any comparable course around. And we also have a very strong tutorial system. So you will have unlimited access really to world-leading researchers who work in the topics that you'll learn about. And so you can catch up on bits of lecture material that you want clarification on or you want further information on. And we also give lots of general information about being a scientist as well. So we have in your personal tutorial system, we have sessions on writing an essay, writing a publication and a lab write-up, how to give a presentation, for example. All of these more general skills that you really need going forward to be a successful scientist. And another huge advantage of being at UCL is that you will be surrounded by world-leading researchers in our department. And you, right from week one, you will be given lectures and you will be taught the material by the very, very best people in the world who could be teaching you these topics. We produce a lot of world-leading research, and we have a lot of some of the most esteemed researchers in various of the fields that we choose to study. And all that leads into the degree being incredibly well regarded by employers, both because you've come from UCL and because you've studied pharmacology at UCL. And it really does open doors when you come out the other end and you're looking to either do further study, for example, with a PhD, or go into the pharmaceutical industry. Okay, so what's the structure of the degree? Well, we have a BSc and an MSci option, so I'll go into the difference of these. So each degree year is broken down into what we call 120 credits. And modules can either be large, so 30 credits taking up a quarter of your year, or small, just 15 credits. In terms of how the weighting of your years builds into your degree, so in your first year, where everything will be a bit new and you'll get used to in many cases living away from home, we start you quite gently, and the weighting of the years is 1 to 3 to 5. So just 1/9 of your degree will rest on that first year, and then 5/9 in your third year. If you end up doing an MSci, then the weighting of those years is 1 to 3 to 5 to 5. Okay, and so what do you start learning when you come here to do your pharmacology degree? Well, starting at the bottom of this list, we have statistics. So it's incredibly important when you do science to be able to prove to other people what you have done. It's really important when you read other people's work to be sure that they have done their experiments rigorously and all their findings are absolutely robust. And so understanding statistics is incredibly important. This is a focus course focusing on the kind of statistics that you're likely to need when you do pharmacology experiments. We also talk about cells and development. So the structure of cells, the nature of cells, the organelles of cells, exactly the kind of communication that happens between those cells and the way that they change during development. So a lead into some of the important anatomy studies that you'll need to understand. Another really, really important thing is biochemistry and molecular biology. So biochemistry in general relates to the function of proteins, whereas molecular biology relates to the function of RNA and DNA. And understanding the exact way that proteins work, RNA, DNA work is again really, really important, in particular for modern pharmacology. Then the first of the two big units you have in your first year, chemistry for biologists. It's really, as you can understand, it's really, really important for pharmacologists to understand the chemistry of the molecules that you use, the chemicals that you're putting into the body to try and produce changes. And of course, those changes will involve changes in the physiology, understanding what happens how does the heart work? How does the brain work? How does the circulation work? What can we hope to try and change when we give a drug to a patient? And what new treatments are still out there that need to be discovered? All of this requires a detailed understanding of the way the body works. And then finally, I've saved the best till last. This course here, An Introduction to the Mechanisms of Drug Action, is your first introduction to hardcore pharmacology. This is really what the degree is about, understanding how drugs act on the body, the kind of mechanisms they use, both on the cellular level and on the tissue level. Okay. And so that gives you a foundation across a whole range of topics that you will need to understand for later years. And then in year 2, this is where this is like the meat of the pharmacology degree, and in particular this course here, so general and systemic pharmacology. Now this is a 30-credit course, so it takes up a quarter of your second year. And it is incredibly important part of your education because it covers the vast majority of classes of drugs and the conditions that we can potentially treat with drugs in the state of the art at the moment. So these can be, for example, drugs affecting the brain. So things like treating Parkinson's, treating Alzheimer's disease, treating epilepsy, things like anesthesia. They can be affecting other parts of the body, like drugs that affect the heart. Drugs that affect circulation. Other drugs, for example, drugs that affect hormones. And there can also be drugs that are proteins or RNA, DNA. So vaccines, for example, are RNA-based drugs. And understanding the nature of all of these drugs and what they can hope to achieve is the main topic of this course. And it's all taught material, it's all lecture material, because there's so much that there is to know. But what that means is that in parallel with this, we have another 30-credit module called experimental pharmacology. And in contrast to general and systemic pharmacology, this course is all about experiments. This is all about getting in the lab. You have various tissue preparations that you'll use, and you apply various different kinds of drugs, understanding the way that these drugs can affect the responses of those tissues. And it's all about the experiments. So there are little sessions where we talk about the design of experiments, the right of experiments, but the main point of this course is just to get you in the lab, get you familiar with everything that you may require to start to perform your own experiments. And so that is half of your year. That's the main part of pharmacology. And then we start to explore for the first time certain kinds of specialities. So neuropharmacology is a real strength and a real specialism of this degree. And so we introduce you to the structure and function of nervous systems. Immunology is something that we're also very strong in. For example, inflammation, inflammatory mediators, and so on. In actual fact, of the 50 new drugs that were approved by the FDA last year, 13 of them are actually based on antibodies. So you can see that immunology has already kind of spilled over into more general treatments for various conditions, in particular cancer. And then you get the first of your potential elective options. So if you're really interested in proteins and protein structure, you can take this essential protein structure and function course. Or if you're more interested in DNA and RNA and the way we regulate gene expression, then you may decide you want to choose essential molecular biology. And you also get a bit more of a free choice as well. So you can, of course, take both the protein and the DNA courses I just mentioned. Or, for example, if you want a bit more neuroscience, you can choose cellular neurophysiology. Or you can even branch out a little bit further with program tutor approval. If you're doing very well in your studies and we don't think you'll be too distracted, you can even do something like learning a language, for example, or helping to develop language skills you may already possess. Okay, so you just start to get a little bit more control over exactly what you study in year 2. And then in year 3, this is where things can become much more specialized. So there is just a single compulsory module, which is molecular pharmacology, and that covers some very much more advanced pharmacology in terms of its mechanisms, in terms of particular kinds of drug. And along with that, the main part of your third year will be some kind of research project. Now, this could either be a laboratory research project, or it could be, which it would be worth 45 credits, or a library research project that would be worth 30 credits. Now, in terms of the research projects, these are not 100% guaranteed, though we will try and make as much availability to do these as we can, and I'll come on to some of the ways that we're trying to increase our capacity towards the end. But you may, of course, decide you'd rather not do a laboratory project, you'd rather do a library research project, in which case you could take on, with the supervision of one of our world-leading researchers, a really cutting-edge topic in pharmacology. You know, which papers have been published last year, which papers have already been published this year, and you get to really focus on a cutting edge aspect of pharmacology, driving forward your own interest, driving forward your own research, and getting right on top of a really kind of cutting edge, at the forefront kind of topic. And so once you've had these research projects and the compulsory module, you've got half your third year, or even more potentially, to take on a broad range of cutting edge other courses. These will typically be 15 credit specialist modules, and these will really allow you to follow interests that you've already developed in the first couple of years. They'll also allow you to explore new fields if there's areas you haven't yet discovered, but you'd really like to do some more research into. And together, this really helps to shape your early career. This is where you really kind of advertise to future employers. This is what I'm interested in. These are the skills I have. This is the knowledge I have that sets me apart from other candidates for that potential position. And there are too many options really to go into, but these may be more neuroscience options, drug design options, overlap into neighboring departments in terms of cell biology, in terms of genetics. Lots of these options are available, will be available to you. And also there was probably courses that will be in existence when you reach year 3 that don't yet currently exist, because we're constantly evolving our output. We have new lecturers that will be joining us this year, and depending on their specialities, we may well end up concocting completely new courses that we haven't previously run. Okay, so that's year 3, and that could potentially be it for your degree, or maybe you would like to either apply right now or potentially transfer to an MSci. And this 4th year, the main point of this 4th year is to do an extended laboratory research project. So these would either be 60 or 90 credits, so potentially taking up to 3/4 of your first year, of your 4th year. And this will be where you can look around our department and neighboring departments and really find an area that particularly interests you, and then try and get into that laboratory to carry out a related project to what they're already working on. And if you have 90 credits, for example, then that's pretty much like the first year of a PhD almost. You really are spending the majority of your time in a lab, really perfecting the techniques that get used in that lab and trying to find out new things about that field. And obviously, once you've taken that research project, then you have between 30 and 60 credits, depending on which version you took, that would again be part of the specialist courses that you can also take in the 3rd year of the BSc. And one other thing to mention is there is the possibility of a year in industry where you can build real world experience in one of the many international, multinational companies that are based somewhere in the UK. Now, this is not something you do alongside your degree. This will extend your study by 1 year, and typically in the BSc, this would be the 3rd year out of what then becomes 4 years, or for MSci, it would be the 4th year out of what would then become 5 years. And these places are not reserved for UCL students, but we will support your application. So they put out general applications, and if you want to apply and you're happy to take that year out, then we will support you in your application. We don't guarantee a placement, even if you really, really want one. The reason for that is that we need to make sure that the quality of the places that's being offered is sufficient. If everybody was guaranteed a placement, we might need to start putting you in companies that weren't as prestigious, that weren't necessarily the right place for you to go. So as long as these places are advertised and we're happy that they're suitable, then we will definitely support your application. But you will be competing with students from all over the country, potentially all over the world, for these relatively few placements. Okay, so typically our entry requirements, these do tend to fluctuate, but our standard minimum offer at the moment is AAB, and this will require for A-levels, and this will require chemistry plus 1 other science. The equivalent with International Baccalaureate is 36 points, and typically we accept between 40 and 45 students onto the course each year. And we had, I believe, over 600 applicants last year. So it really is a very prestigious and very, very competitive course to get onto. The university would love us to be able to offer more places, but we simply just cannot, because we're so insistent that we have so much hands-on experimentation. That does limit the number of students we could have, because if we had many more students, we'd have to dilute down the amount of time you actually get in the laboratory to do experiments. Okay. And so if you come to UCL, well, what happens after your degree in pharmacology? Well, the majority of our students go on to do some kind of further study, whether it's a master's or a PhD. But there's also the option to do other study, like even transfer to or then move on and do a medical degree. And it may be possible that you can do a slightly shortened medical degree if you have completed a BSc in a related topic like pharmacology. The next options on this list are all related to the pharmaceutical industry, and the pharmaceutical industry is really is what our degree is really set up to feed into. And so, for example, if you wanted to do further study, you could do like patent law. So patents and intellectual property are an incredibly important part of the pharmaceutical industry, because if they can't protect their discoveries, they can't protect their new drugs, then they can never hope to recoup the cost it takes to develop them once it's actually out there. And they need people with pharmacology like training to do this aspect of the legalities as well. Of course, you don't necessarily need to do further training. You could go straight from your degree into the biotech industry, the pharmaceutical industry, for example, and not just in terms of research and development. There are other things like sales, for example. When a drug company develops a new drug, they need to get the word out there to all the clinicians, because if the clinicians aren't prescribing it, then there's no point them, they're not going to make their money back. And so they need people who understand the way drugs work to go out and talk to the doctors to try and persuade them to prescribe that drug. There are other things like regulatory affairs. So this is things like making sure in control of drug safety, in clinical trial design, making sure the drugs are affordable, making sure the manufacturing is robust, all this kind of thing. All that, again, really, really important in terms of actually getting drugs onto the market and into patients. And then, of course, you can go somewhere slightly directly away from science. So, for example, going to the city, the financial sector, they need to understand these industries so they know which industries to potentially invest in. So you can go and use your skills in that regard. Or, of course, more kind of regulation directions for this kind of education, which would be going into some kind of teaching, or maybe going into science publishing or medical publishing. Again, a very, very common destination for science students in the UK. Okay, so if you want to know more about where pharmacology might be able to take you, the British Pharmacological Society have a very useful webpage, which you can see the link for there, which will allow you to understand both in a bit more detail what I've just gone through, and also many other potential destinations as well. Okay. So that was just kind of an overview of the degree. What I want to talk about now is just give you an example of the kind of research that goes on in our department and goes on around UCL pharmacology and quite a few places. In particular, this is the work that goes on in my laboratory. And the reason why I've called it Your First Discovery is that I'm in the process of setting up a lab that will allow third-year BSc students will allow us to give laboratory projects to many more third-year BSc students. And I'm going to tell you a little bit about the work we've been doing and the kind of work, therefore, that you will hopefully, if you're interested, be able to do if you are to come to my lab. I should also make it clear that this is not the only project that you might be able to do if you come to do a BSc laboratory project. There are other places you can go as well, but I should also make it clear that there are no guarantees. Even with this new capacity, we will necessarily be able to give laboratory projects to everybody. So, so as I mentioned, we are big on neuropharmacology in our department, and the nervous system is made up of trillions of nerve cells or neurons, and these cells are crucial for transmitting electrical signals through the brain. So I'm just going to give myself a little laser pointer here so I can point out what there we go. So this is the cell body of a neuron, colored in blue there, and that is that the blue is highlighting the DNA. So that is where the nucleus is. That is the cell body. But neurons also have all these amazingly beautiful kind of processes here in green, where they send out little tendrils to communicate with other cells in the vicinity and to pass the signals along themselves. And as well as all these processes that they have themselves, if you look at all these little red dots that are dotted around everywhere, these are the junctions between neurons, so where they meet each other. And in general, the communication that happens in the brain along neurons and within the cells is largely electrical, whereas these synaptic junctions between the neurons are a very brief chemical where a chemical signal passes. And in terms of neurons, these chemical signals are called neurotransmitters. And in particular, what we work on in our lab is a neurotransmitter, an excitatory transmitter called glutamate. And it carries the vast majority of excitatory signals between nerve cells in the brain. Okay, so how do they turn this chemical signal back into an electrical signal? Well, glutamate receptors, as with all receptors, are membrane proteins, and receptors can bind things like hormones and also neurotransmitters like glutamate, and this converts an extracellular signal into an intracellular signal. Now, in terms of the receptors we look at, these are a particular class of receptor called ionotropic receptors. And what they do is they bind glutamate here in a region of the protein called the ligand binding domain. And this communicates with a region called the transmembrane region that's in the membrane. And the point of these glutamate receptors, which are 1,000 amino acids long, well, they come together in groups of 4 and they surround a pore in the membrane that's called an ion channel, and it allows ions to pass through, and that is the electrical signal. Now, as in all modern pharmacology, and glutamate receptors are no different, we know an awful lot now about the 3-dimensional structure of the proteins that we want to examine. And so we can understand an awful lot about the exact ways that each of these little parts of the protein interact with the drug, and what kind of drug, what kind of molecule is likely to produce what kind of change within the cell, within the neuron. And so because I've been in this laboratory researching these proteins for around 20 years, the laboratory itself has been around for over 40 years. And because we're at the forefront of our field, as so many of the groups at UCL are, we were reached out to by a family-led foundation called the Kjorgren Foundation. And basically, this was a foundation set up by families who had children who had de novo genetic changes in a glutamate receptor gene. And what are de novo changes? Well, we are 50% our mothers and 50% our fathers, but we're also 0.000001% error. And so there are around between 30 and 50 genetic changes that are present in you that weren't present in your parents. And the vast majority of these are completely harmless. They don't really do anything. We don't really notice. But if you get the wrong change in the wrong place, then it can be devastating. You can have out the blue a child who's very, very sick. And that was the case with Kjorgren. They were interested in a particular kind of glutamate receptor called an NMDA receptor that's encoded by the GRIN genes, GRIN1A, GRIN2A, and so on. What they also found more and more commonly was that they were also being contacted by patients who had variants in the GRIA genes. And these are the genes that encode for a different class of glutamate receptor called AMPA receptors. And AMPA receptors are what we specialize in. And so they knew this and they reached out and asked us if we wanted to help. And we did, of course. And so as a result of our work, amongst other people, of course, in 2021, their logo went from being Kjorgren, researching the GRIN disorder, to now also researching these other kinds of glutamate receptor disorder as well. Okay, so what can we actually do? How can we actually use pharmacology? How can we use our laboratory to actually help this foundation? Well, the first patient that we came across was a young boy who presented with episodes of back arching and vomiting at 6 months, which was very unusual. And by the time he was 12 months old, he was having regular seizures, an example of which is seen here on the right. And this led to him being on a number of anti-seizure medications, but despite this, he continued to have seizures. So at this time, his situation became more and more severe, so global developmental delay became more obvious. He had worsening GI symptoms in terms of his ability to eat. and he was diagnosed with failure to thrive because his body weight was just getting so low. And after numerous tests and numerous efforts to try and improve his situation, eventually he was found a genetic diagnosis, which was a de novo variant, so not seen in either of his patients, in his parents, in GRIA2, which is an AMPA receptor gene. And it was this particular change here, alanine in position 643, in his case was a valine, and it was deemed to be lightly pathogenic. And having looked at the position of this variant, we were really sure it was likely to be pathogenic as well. So in general, the way you can work out which bits of a protein are the most important are the bits that stay the same. So when there are lots of different proteins in a family, you can tell which are the most important bits because they stay the same. There's genetic drift in other particular regions that aren't so important, but the key bits are the same. And this valine change was found in a region of the glutamate receptors that was completely conserved. And so we knew this, but this was important. And the reason it was important was that the position of this amino acid was found just at the gate region of the receptor. So this is where glutamate binds to the receptor and tells the ion channel to open. And so this alanine was found right at the part that when the glutamate binds, the channel opens, and when the glutamate goes, the channel closes. And this is absolutely crucial for the speed at which receptors work and their ability to open and close. So we were really sure this was likely to be the cause of his problems. And so we used a technique called electrophysiology. And this particular technique that we used was to put the de novo variant form of the receptor into a cell. And then what we did was we grabbed a tiny little bit of the membrane of this cell and then removed it from the cell. So we had a really tight, really small bit of membrane, really tight electrical control over this membrane. And what it allowed us to do is to trick the receptors into thinking they were at the synapse using this kind of cool technique here. There's a little cartoon. And so this little patch of membrane here is this little patch of membrane here on the end of the electrode there. And what we did was we jumped it between two solutions, a control solution without glutamate, so nothing to activate the receptor, and then jumped it on this side to a receptor that had 10 millimolar glutamate. And you can see what happens to the receptor when you go from a situation with no glutamate to high concentrations of glutamate. This downward deflection is the electrical current that passes into the cell or into our bit of membrane. And it happens incredibly quickly, but then it closes incredibly quickly as well. This is the way that AMPA receptors work. They're really, really, really quick. Now, when we did this technique on the variant, you can see in this particular trace here, so the healthy form of the receptor, the form of the receptor that you and I have, is very, very quick. So you apply glutamate for 1 millisecond, the receptor will activate very quickly and it will close again within 1 or 2 milliseconds. But what we found in purple here with this variant is it activated and then it didn't close as quickly. It stayed open for a little bit longer. Okay, and this kind of provides an explanation as to why the patient was having seizures, because even when he was trying to pass a very, very quick short glutamate signal, his receptors were holding on to the glutamate for too long, and they were leading to much too large a signal. And so it's no wonder, given that glutamate is the predominant excitatory transmitter in the brain, having these elongated glutamate receptor responses led to massive overexcitation and led to him having seizures. Some other tests we could do as well, and these are fitted with the Hill equation. So that was A.V. Hill, was an alumnus of UCL 100 years ago, who invented important aspects of pharmacology. And you can see that the sensitivity to glutamate was greatly increased with the variant, and so less glutamate was required to produce the same response. But crucially as well, perampanel, which is a blocker of the glutamate receptors, was also effective and could also inhibit this overactive form of the receptor. And so what that meant was that we were able to communicate back with the clinicians who were looking after this patient and say, perampanel is not normally given to patients that are under 4 years old, but we had a very specific reason why it was very much worth a try. And because his medical situation was so serious, they were able to give the patient this drug. And it was really, really transformative for his situation. So you can see here in the months before he started perampanel, he was having up to 12 seizures a day, even given a high dose of this anti-seizure medication. He was unable to control this seizure activity. But when he started perampanel, there were virtually no seizures. And he was even able to stop all these other medications and keep his seizure load down. And so potentially this was to be expected because this was an anti-seizure medication. It wasn't a conventional one to give for a child so small, but potentially it wasn't a surprise. But what was really cool was that because we'd got to the real cause of his disease, not just the cause of his seizures, he then started to have, these pictures are shown with permission, I should point out. He then started to have lots of other huge gains in his development. So his failure to thrive resolved. He put on a load of weight. His new physical milestones were reached. So he was able to sit up for the first time that he wasn't able to do before. And really importantly, because some of the symptoms associated with these disorders are quite psychiatric, his social interaction increased. He started making eye contact with his parents and started trying to even say a few words. And so by using pharmacology, we were able to identify the exact right treatment for this patient. And he had a much stronger result, a much better clinical result than could certainly have been expected given how he was at 12 months old. And so let's go back to your first year. So we needed to statistically show that this was important. No clinician is going to give an unconventional drug to a child unless you can conclusively prove there's a reason why it's a good thing to try. Clearly, this was important for cells and development. This is a neurodevelopmental disorder. So understanding how development worked was really important for us to help understand his situation. The biochemistry and molecular biology are, of course, important. The genetic tests that actually told us that it was an AMPA receptor variant was the cause of his problems. And then the chemistry for biologists, we knew the exact side chain that had changed. So we knew the exact tiny bit of chemistry that was different between the wild type receptor and the variant receptor. And again, that's stuff that you learn about in your first year. And of course, the physiology is important as well. These are electrophysiology experiments. Understanding the way this bioelectricity works was absolutely crucial in coming up with the correct answer for this patient. And of course, pharmacology. So the introduction to the mechanisms of drug action. This is a non-competitive antagonist drug that we applied in our system. And given everything we knew about it, then we knew it was going to be it was going to be a really good thing to try for the patient. And so it proved. So when you come to UCL, right from year one, you are learning about all of these really, really important fields, all these really, really important areas that can then be directly applied to experiments that you can potentially be performing yourself. And yes, of course, you can perform yourself, you can get involved. So we're still working closely with clinicians worldwide, and new cases of these glutamate receptor disorders are being identified all the time. And there are hundreds of patients with glutamate receptor genetic variants that still need a diagnosis. And so one option for your third year laboratory project will be to come and work in my laboratory to help contribute to this work, to help better understand what is still a very rare disorder, to help identify precision therapies, and hopefully, and you may of course take your pharmacology degree and go and solve some of the biggest problems that face mankind, whether it be Alzheimer's or addiction or antibiotic resistance or anything like that. But on the way there, you get the chance potentially to help one little person who may very well have an answer that they need to be given to have their best care. And so this could be a project you do in your third year as part of the BSc or a fourth year as part of the MSci. And of course, you also get many equivalent projects, many similar projects to choose from all over our department. And just one very quick thing to finish. We just had a paper published, not on this patient, that one was from 2022, but on another few variants. And as well as my name and a couple of other people in the lab, you can see Xinyao, who's also on the call today, who, as part of her work in our lab, has already got her name on a paper. And this is the real currency when it comes to CVs, when you're trying to impress future employers, you're trying to impress pharmaceutical companies. Having this kind of detail on your CV is really, really, really important to help open even more doors than just UCL pharmacology can do. And Xinyao has already got a position for the next year working up in Cambridge, which, as we've established, is an inferior pharmacology department. Don't ask me. US World Rankings. But nonetheless, it's quite a good place to go. So well done to her. And our objective is to try as much as possible for the work that goes on in our lab to ultimately be published, to be as available to as many people as possible to help them understand. So, for example, if the same variant crops up in a second patient, the fact that that information is out there will allow them to instantly treat that patient better. Okay. So those are some options potentially going forward. Okay, so sorry, I've talked a little bit long there. I get very excited when I talk about glutamate receptors. But here you can see, again, that's that link to the Careers in Pharmacology website and the British Pharmacological Society webpage. Yes, and I'll just ask any questions. So as I said at the start, we have some of our current students here. So if you have a day-to-day question about what it's like to be a student at UCL, feel free to put your hand up and unmute your microphone and ask the question, or you could just drop it in the chat. But we look forward to answering whichever questions you have. Thank you very much. So I'm going to fill in the chat. Where's the chat, please? Okay. How busy a degree is pharmacology? I guess this is so typically it depends a little bit on it will vary week by week. You know, sometimes you'll be in a group that's doing a lab practical, sometimes you won't. But typically the contact hours are between 10 and 15 hours. And that will be, by contact hours, we mean lectures, we mean tutorials, lab practicals. And the general expectation is that you will work between 2 and 3 times longer than that in your own personal study. So that would be catching up on lectures, reading around the topic. Potentially, you know, we'd like to give you the confidence to read the most cutting edge information about a particular field, so you can start to ask your own questions and start to develop your own understanding beyond just what we teach you. And of course, you have your personal tutor there who will be always at your disposal to ask whichever questions may come up. Okay. What advice would you give to someone who is deciding between pharmacy and pharmacology? I would say that if you do a pharmacy degree, you can't really then go and be a pharmacologist. Whereas if you do a pharmacology degree, you will potentially have more of an option to be a pharmacist. But strictly speaking, as I said at the start, pharmacology is a science. It's the point of training to do pharmacology is to discover stuff, to help develop drugs. Whereas pharmacy is typically a vocation where you help to dispense drugs and give, you know, obviously you need to understand an awful lot about all the drugs that can be prescribed. But you won't end up doing typically research in a laboratory like you will with pharmacology. So my own preference, of course, would be to study a science and to study pharmacology. Supercurricular activities. So, okay, I guess I could take that question 2 ways. So if you're thinking about things that you want to do before you potentially come to university, there are lots of expose yourself to lots of interesting things that interest you, basically. So if there's a particular disease that you're interested in, look at all the treatment options available. What still needs to be discovered? What is there that can be improved about that? Think about techniques that look really cool, things that you might be really interested in. And again, that can help foster your interest and help come up with new questions. If I take the question as in what can you do extra while you're at UCL, I might kind of reflect that one onto our students, members of the panel, if they're interested. Is there anything in particular outside of lectures and the standard contact hours that you've worked on, you found were helpful for you at UCL? Any clubs, societies, and so on?
Sorry. Pharmacology Society. There's a Pharmacology Society at UCL. I think Jinyao is a committee member of Pharmacology Society, so you can join that.
Yeah, that's just I want to talk about we have the departmental society which is called UCL Pharmacology Society. We organize lots of different talks. Like sometimes we invite professors working from college area, or sometimes we have the people working in some big pharmaceutical companies, and they come to give a talk. Yeah, there are lots of exciting opportunities. I personally also started a project which is called the newsletter, and we do this mostly to talk about some new published paper and some news about the pharmaceutical companies to help you to know some to help you to build your careers and think about what is going on for our research. Yeah.
So a question, any specific books that you'd recommend a prospective student looking to get into pharmacology? Well, the kind of the Bible of pharmacology, really, certainly the textbook that we build most of, that has been the foundation of pharmacology teaching over the last 10, 20 years, is a book called Rang and Dale. It's a textbook. It's broken down very, very nicely into the certain easy to digest kind of chunks. And so there's online versions if you don't want to carry. It's a pretty chunky thing, so you probably don't want to be carrying it around in your backpack. But that would be something to really get across pharmacology. And other than that, as I mentioned before, just kind of keep your eyes and ears open. You know, when there are science pieces in the news very, very often. You know, what is the brand new drug? You know, for example, Ozempic, for example, is probably one of the biggest drugs of the last 10 years. Now, we do certain research at UCL on neuropeptides and appetite and digestion and so on. So trying to expose yourself, not just from textbooks, from all manner of sources, to try and really just see what really captures your attention. And in terms of your application, putting that kind of interest and demonstrating that you've done a little bit of extra reading yourself and you've really dug down into a topic, that will play really, really well when your cover letter is going to be looked through. And that is the first kind of triage we do, because there are so many applications. And so getting that right, demonstrating that you've found an area of pharmacology that is of particular interest to you, is kind of really important. So next question, would pharmacology be a good start to pursue a career in drug discovery? Would it be helpful? Oh, 100%, yes. I mean, one of our most popular third-year courses focuses on drug discovery, actually. And the people who run that have very close links to certain industrial placements as well. And so, yeah, understanding the way, I mean, for pharmacology, you need a drug and you need a target. And that's what pharmaceutical companies are all about. And so understanding the particular ways that drugs can modify particular kinds of target is incredibly important information and is highly valued by the pharmaceutical industry. So yes, this is more than any of those other kind of biological sciences, whether it be physiology, anatomy, this is really on a fast track to that one particular industry, the pharmaceutical industry. So yes, it would definitely be helpful. Yeah, so I think possibly I covered that there's a question about personal statements. And I mean, yeah, basically you want to demonstrate that I mentioned that you have 10 to 15 contact hours, and then you also need 2 or 3 times more. So demonstrating you're the kind of student that will go out of their way to discover and read extra things that you in particular find interesting. There's no right or wrong answer to this, but just demonstrating that you've gone and you've discovered something new, you've found something that interested you and you've really got your teeth into it. That is that will definitely really help your personal statement and really help you to stand out, and hopefully help fostering a real interest and love for pharmacology before you even get here, when of course you will have a really strong love for pharmacology for the rest of your days. So okay. Does the course material change as the new research comes in? Absolutely, yes. We are very keen to make sure that when we talk about some condition or other, whether it be, I don't know, name whatever you want, whether it's cancer or whether it's, Parkinson's disease, or whether it's antibiotics, or whether it's some kind of inflammatory disorder. We don't want to be telling you about the drugs that were discovered in the 1960s and decided and found to be unsafe in the 1980s. We want to be telling you about the things that actually are the most effective things right now. And so, yes, we are constantly in a state of updating our degree programs to make sure you get the best information, basically the cutting edge information about the drugs we use and the techniques that are used to discover them. No, there is no entry test or interview required for admission to the pharmacology course. It's done by a written application through the UCAS system. So again, your personal statement is important. Clearly, you need to have demonstrated with your predicted grades and your academic career to date that you're going to be a strong student, but there's no additional things that are required. Yeah, good question. What is the lab environment like? Does it get repetitive or mundane? It depends on the laboratory. It will never be mundane in my laboratory. I can absolutely 100% guarantee that. And Sinyo knows, because she's been with us for the last year as well. Yes, I mean, potentially it can be. I suppose it depends on the first time you do an experiment. In general, it goes wrong. The second and third time you do an experiment, it probably works, and it's like really exciting because it's the first time you've done it. But going back to the material on statistics, to prove what you found is real, you need to go and do it, 6 times, 8 times, 10 times. And increasing those numbers, I personally find really fascinating. You know that every time you do an experiment, even if it's a repeat of an experiment you've just done, it's making your findings more robust. It's making you even more certain about what you've already discovered. But I suppose it's possible you might just be, you want a new technique every week, and you say, oh, I've done electrophysiology now, I want to do super resolution microscopy, or you've done that, you want to do some kind of CRISPR modification, or you want to do some kind of animal experiment or something. And the good thing about UCL is that we have so many of these cutting edge techniques going on. It's not impossible that you could kind of flit around and come up with a reason to try a different technique. But in general, you will hopefully be super excited about the project that you get stuck into, and you don't need to worry about getting bored. Oh my goodness, the questions are still coming in. So, a West Ham fan has said, I'm sorry about that. Okay, next season. What would make an applicant stand out in their personal statement when applying for this degree? As you said, there are limited spaces available. Yeah, I will just go back again to the fact you need to demonstrate that you are more than just the student who sits quietly in class and takes notes and then looks at the notes and then regurgitates them in the exam. You've got to demonstrate that you've got a bit of adrenaline about your interests. You've got to go and find something. You've got to, like I say, if you see an article in a paper, you see a drug that's been discovered, or even a drug that's suddenly been proven to be unsafe or something like that that's in the news, go and read up about that. Go and, okay, so that's not working. What could be the next thing? What other options are available? And yeah, demonstrating that level of extracurricular interest will go beyond your ability to get all the right answers in the exam, because ultimately the way you learn at university is different than the way you learn at school. The way you learn at school, they give you all the information you need. You need to try and remember it all, and you need to try and remember it in exam, and they might kind of dress it up and try and disguise it in the question. But you don't need to, you just need to do really well what they give you. At university, you've got to go further. You have to take the material that's there in the lectures, and then you've got to try and work out, well, what's next? You've got to go and read extra. And when it comes to your exams at the end of the year, if you get all the answers right, that's brilliant. but then you also have extra statements to make, and you know, also, and I'm marking these scripts going, oh, I didn't teach them that, or they've gone and they found it out themselves. And you need to demonstrate that you're that kind of student even before you arrive, and that will really make your personal statement stand out and make you, you know, make it more likely your application will be successful. Would work experience in a lab help my application? I guess so, yes, because I mean, it's not, I guess it's slightly harder when you're at an A-level stage to get that kind of experience. But again, that shows that you have an interest that is beyond just reading the books. You have an interest that will potentially, you know, you've taken the time, you've taken effort to go and actually experience something outside of your schoolwork. And so, yes, that definitely would be something that would be taken into account when they look at your CV and so on. Would pharmacology possibly help you get into the anti-doping industry? Oh, I love specific questions like that. And yeah, of course. Yeah, I mean, whether it's, I'm not completely up on anti-doping these days. I don't know what I think in general, they're taking it anyway and then saying they ate some contaminated meat or something. And then eventually after like 9 months in court, they just get to carry on as if they, you know, as they were originally. So yes, catching the, if you can catch the drug cheats for me, so I can believe in my sporting heroes more convincingly, then yeah, 100%. And of course, pharmacology will. I mean, you, for example, in the second year experimental pharmacology, the second year systemic pharmacology course, you learn about hormones, for example, and so testosterone and all these other kind of systems that can be modified by such things as the antidote drug cheats. Yes, so yeah, why not? Absolutely. Oh, a question for the students. Are you seeing this one? So what is something you wish you knew about the course before you started? Okay, excellent. I'll make notes on this one. Any contributions?
I think they've answered in the chatty, and
oh, sorry, my mistake. I didn't go ahead. Okay, so chemistry, yeah, very important. Yes, getting on top of all the drug names. It's important to know the types of drug and the way they act, but of course, you need to give specific examples. Yes, excellent. All very good. Okay. So if someone is doing summer school work experience at UCL this summer for medicine as pharmacology was an option, would that still support my personal statement? Yes, absolutely. Yes. Again, you just need to be active. You need to demonstrate. You need to be able to evidence and quantify your interest in this subject by going out and discovering something. And yeah, 100% that will help. Yes. Would it be hard to catch up if I didn't study biology? Well, I'm going to say no, actually, because it's not a requirement. So there will be certain aspects that will come up that you would have covered in biology, but because we don't expect you to have taken biology, it will be covered. It may be covered in a much more focused way to answer the specific aspect of pharmacology that we're teaching you. But yes, no, it would definitely be. It wouldn't be a problem to catch up. If you wanted to catch up, of course, then actually I would say quite a good way of doing it is just buying an A-level revision guide. And then you will have everything that you would have come up in a biology A-level in a quite a neat kind of packaged form. And you can dip in and out of that during your summer holiday. And make sure that if there are any areas that you really don't understand, you're not familiar with at all, then yeah, you can catch up. Okay, I think we've, oh no, one more. Oh, sorry, no. Okay, so if there are no more questions, then yes, please do. As you can see in the chat there's some details. We do have some in-person open days as well. So if you'd like to actually see the campus, if you would, I'm hoping, I need to double check I can do this. I have a little pharmacology experiment lined up for the open day. So be sure to catch that. But if there are no more questions right now, then there was one more question right now. During that week, will you be able to doing? Ian, can you help with that? Yeah, sorry, this is my first year being admissions tutor actually. So I've only been admissions tutor since January, and I'm not sure if there are any other sessions elsewhere in the year.
I'm afraid those will be the only 2 in-person undergraduate open days that we're running this year. But I've put a link in that you could take a virtual tour of our campus, and I've put a link in the chat for that if you wanted to just get a sense of what our buildings and laboratories and spaces are like. Take the virtual tour.
Can we include taster days or meetings like this in our personal statement? I mean, I guess so. If something happens while you're looking around the teaching lab that completely inspires you, then yeah, why not? I guess yeah, I don't have any. I mean, yeah, it may well be that you get to see a particular laboratory, or get to see a particular part of UCL, or you learn about the history of UCL, a particular, a lecture theatre that's named after a particular person that you want to go and find out a bit more about. That would yeah, that would be fine as well. Okay. Okay, well, thank you very much for your attendance. I may end up seeing some of you in June at the in-person open day. But thank you very much for your time, and I hope to see you in the near future. Thank you. Goodbye.